Introduction. Glaucoma is a leading cause of irreversible blindness worldwide, affecting tens of millions of people. It is traditionally linked to high eye pressure, intraocular pressure, but many patients continue to lose vision even when pressure is controlled. Scientists now think that pressure is only part of the story. Inside each retinal ganglion cell, RGC, the neurons whose long fibers form the optic nerve, a complex energy crisis may arise over years. In this scenario, glaucoma becomes an energy failure disease. If an RGC cannot make enough energy, its axons and connections slowly fail, damaging vision. This article explores why optic nerve cells need so much energy, how aging and stress may starve them, and what researchers are trying, often by boosting cell power, to save the nerve. We'll also connect these ideas to other brain diseases and early experimental treatments that aim to shore up cellular energy. Why retinal ganglion cells need huge energy. Retinal ganglion cells are the nerve cells in the eye that send visual signals from the retina to the brain. They have an especially high energy demand. Unlike most neurons, RGC axons, the nerve fibers, travel a long distance without the usual insulating sheath called myelin. In fact, all along the length of the retina and optic nerve head, RGC axons are unmyelinated. Each electrical signal, action potential, must be actively regenerated step by step, which uses a lot of energy. To meet this demand, RGCs pack in mitochondria, the cell's power plants, along their axons, especially at the optic nerve head where the fibers take a sharp turn out of the eye. The region just inside the optic nerve is mechanically stressful, squeezed by eye pressure and movement, so RGCs concentrate mitochondria there to keep energy up under strain. In short, RGCs are among the most energy-hungry cells. They never stop, and their unique structure means they are built with dense fuel supplies. In practice, this means any problem that reduces their fuel can quickly hurt RGCs. Neurons rely on two main pathways to turn nutrients into ATP, cellular energy, glycolysis using sugar, and oxidative phosphorylation using oxygen in mitochondria. RGCs ride a delicate balance between these, and they depend on continuous delivery of oxygen and nutrients through tiny blood vessels. Even slight disruptions, like slower blood flow or extra pressure, can tip the balance. Glaucoma stressors: pressure, blood flow, and aging. Glaucoma stresses RGCs in several ways, any of which can hurt mitochondria and thus energy supply. Eye pressure and blood flow. Elevated eye pressure makes it physically harder for blood to reach the retina and optic nerve. Imagine pinching a hose. Reduced blood and oxygen supply starves cells of fuel. In glaucoma, this can create brief ischemia-reperfusion injury, a kind of mini-stroke, where blood flow drops and then suddenly returns. During this process, mitochondria produce extra reactive oxygen species, ROS, that act like toxic sparks inside cells. Indeed, animal studies show that high pressure causes a surge of oxidative stress in the retina. For example, when researchers raised eye pressure in rats, levels of glutathione, the cell's natural antioxidant, plummeted, while markers of superoxide, a damaging oxygen molecule, rose in the retinal ganglion cell layer. In other words, high pressure literally starves RGCs and floods them with damaging free radicals. Over time, this chemical stress weakens RGC mitochondria, making them less able to make energy. Aging and NAD decline. Age is the other big risk factor. As we grow older, all our cells lose some ability to fight stress. In RGCs, a key change is a drop in NAD, nicotinamide adenine dinucleotide, a molecule that cells use like currency in energy production. Multiple studies in glaucoma models report that retinal NAD levels fall with age and with pressure. This makes a perfect storm. Older RGCs have less raw fuel, NAD, to run their mitochondria, so they are already close to energy failure. The consequences are clear in experiments. In a mouse study, the researchers found that boosting NAD by giving nicotinamide, a form of vitamin B3, protected RGCs starkly. At the highest dose, 93% of treated eyes had no glaucoma damage at all, even though eye pressure still rose. This shows that simply refilling the battery can nip the damage in the bud. In other work, aging mice given high-dose nicotinamide kept their NAD levels high long-term and resisted vision loss. Conversely, human glaucoma patients have been found to have lower blood levels of vitamin B3 compared to people without glaucoma. Altogether, the evidence suggests that age-related NAD loss tips some RGCs into an energy crisis. Oxidative stress. When cells burn too much, oxidative stress is a term you will hear often in glaucoma studies. It simply means the balance between harmful oxygen molecules, like free radicals, and the cell's antioxidants is tipped so far that damage occurs. Mitochondria naturally leak some reactive oxygen during energy production, and small amounts are normal. But when pressure, poor blood flow, or aging disrupts the system, RGCs generate excess radicals faster than they can clean them up. One review explains, reactive oxygen are essential participants in cell signaling, but when production overwhelms the antioxidant capacity, damage to cellular molecules ensues, a state of oxidative stress. In glaucoma, oxidative stress is seen in multiple ways. Studies have found oxidative modifications of proteins in dying RGCs and loss of antioxidants in the eye's fluids. In experimental models, artificially raising eye pressure causes spikes of oxidative markers in the retina. Oxidative stress itself can damage mitochondria and other cell parts. Proteins, DNA, and membrane fats get shot by these reactive species, making mitochondria less efficient and cells more prone to self-destruct. This is why antioxidants are considered for therapy, see below. By bolstering the cell's cleanup crew, we hope to prevent the energy machinery from self-immolating. Mitochondrial dysfunction and optic nerve damage. When mitochondria start failing, an RGC can't make enough ATP, its essential energy packets. The results are profound. The nerve fiber, axon, can no longer transport cellular cargo, like proteins and organelles, up and down its long length. Researchers describe this as a breakdown of axonal transport. Think of it like cargo trucks stuck on a road because there's no fuel. In glaucoma models, impaired axonal transport is one of the earliest signs of trouble. This eventually leads to thinning of the optic nerve and failure of synapses in the brain, and the visual field loss patients see. Microscopic examinations confirm that mitochondria look abnormal long before RGCs die. For example, in one glaucoma model, the tiny folds inside mitochondria, cristae, become reduced on electron microscopy, signaling collapse of energy factories even before any cell loss. The cells also lose internal structure. In DBA2J mice, a glaucoma strain, RGCs start retracting branches and pruning connections once energy falters. Bursting these processes of energy shortfall and structural damage is a vicious cycle. More oxidative stress impairs mitochondrial function, and bad mitochondria create more oxidative stress, along with activating cell death programs. Thus, by the time clinical signs appear, the RGCs have already lost much of their support. This energy starvation model helps explain why some glaucoma patients, especially the elderly, continue to worsen even with normal eye pressure. Their cells simply can't keep up. Neuroinflammation and the eye's immune storm. Another layer is neuroinflammation. The optic nerve is supported by glial cells, like astrocytes and microglia, that normally help neurons. But when RGCs struggle, they send distress signals that activate these glial cells. At the same time, damaged mitochondria themselves release inflammatory cues. For instance, fragments of mitochondrial DNA can act as danger signals that trigger the cell's immune sensors, e.g. the NLRP3 inflammasome, causing release of inflammatory cytokines like IL-1 beta. Once inflammation kicks in, it further robs cells of energy, it takes fuel for immune reactions, and can directly damage neurons. In fact, a recent review noted that in glaucoma, crosstalk between mitochondria and inflammation accelerates damage. Injured mitochondria amp up immune signals, and in turn, immune signals further drown the cell's power production. Practically, this means that high pressure or oxidative stress in the optic nerve can lead to an immune reaction similar to what we see in Alzheimer's or Parkinson's disease, contributing to a downward spiral in RGC health. While our technology is still catching up in mapping inflammation in the eye, it's clear that metabolic failure and immune activation go hand in hand. Imaging of human glaucomatous optic nerves shows markers of inflammation, and many immune-related genes are switched on in stressed optic nerve tissue. This is an active area of research. If we can tamp down harmful inflammation by protecting energy factories, we might break the cycle of decline. Searching for energy-boosting therapies. Given this energy picture, researchers have begun targeting glaucoma with metabolic therapies. The idea is, if the optic nerve cells are starving, let's give them more fuel or helpers. Here are some promising but still unproven approaches under study. NAD precursors, vitamin B3. Boosting NAD levels has been especially exciting. Nicotinamide, the amide form of vitamin B3, elevates NAD in cells, powering mitochondrial function. In mouse models, high-dose nicotinamide preserved RGCs amazingly well. This led to preliminary human trials. One controlled trial gave glaucoma patients 3 grams per day of nicotinamide and found measurable improvements in retinal signal tests, pattern RG, suggesting better RGC function. Importantly, nicotinamide was safe and did not lower eye pressure. Its benefit was purely neuroprotective. Research now also explores nicotinamide riboside, another NAD precursor with good bioavailability. In a small clinical report, combining nicotinamide riboside with berberine, a plant compound that activates cell energy pathways, stabilized visual fields and nerve fiber thickness over 6 months. These results hint that supporting cellular metabolism can slow glaucoma, but larger trials are needed before any recommendation. Antioxidant supplements. Strengthening the cell's antioxidant arsenal can indirectly support energy. Various substances are under investigation. For example, coenzyme Q10, CoQ10, is a cofactor in mitochondria that also acts as an antioxidant. In rats with induced glaucoma, CoQ10, often given with vitamin E, reduced neuron damage and cell death. Other compounds like alpha-lipoic acid, vitamins C and E, resveratrol, omega-3 fatty acids, and hesperidin, a citrus flavonoid, have shown protective effects in lab experiments. Some eye drops and nutraceuticals enriched with these are being tested for glaucoma, but the clinical evidence is still slim. A non-invasive one, a dietary antioxidant pill, showed increased antioxidant capacity in small human studies, but we await proof that this slows vision loss. Overall, giving extra antioxidants is a low-risk idea that may help flush out reactive molecules. Metabolic support and diet. More broadly, lifestyle factors can influence cellular metabolism. Regular exercise and a healthy diet, especially a Mediterranean-style diet rich in fruits, vegetables, nuts, and olive oil, improve mitochondrial function in the brain and retina. Ensuring adequate intake of micronutrients, B vitamins, vitamin C, E, selenium, etc., supports the body's own antioxidant systems. In theory, very low-carb, ketogenic diets or mild fasting could switch RGCs to burn ketones, an alternative fuel, and strengthen their stress resistance. Experiments in other nervous system diseases suggest potential, though this is not yet established for glaucoma. Some small studies even combine metabolic fuels. For instance, taking nicotinamide together with pyruvate, a simple energy molecule, briefly improved vision test results in open-angle glaucoma patients compared to placebo. These approaches are still exploratory, but they highlight that what we eat and how we live might modestly affect retinal energy balance. Pharmacological and gene therapies. Beyond natural compounds, certain drugs and genes are being explored. An example is brimonidine, a widely used glaucoma eye drop, which in animal studies showed neuroprotective effects independent of pressure. Affected eyes on brimonidine lost vision more slowly even when pressure was not high. Its mechanism might involve mitochondrial tolerance, though it's not fully understood. On the gene side, researchers have engineered mice to overproduce the enzyme NMNT1 that makes NAD. These mice showed remarkable resistance to glaucoma damage. In one experiment, mice that had both the NMNAT1 gene therapy and nicotinamide nearly completely avoided vision loss. These are very early-stage ideas, far from clinical use, but they underscore a proof of principle. Directly boosting the neuron's energy machinery can protect the optic nerve. Experimental strategies. More futuristic ideas include transplanting healthy mitochondria into the eye, stem cell therapies, and even light-based treatments that stimulate cellular repair pathways. A recent review listed everything from mitochondrial transplantation to low-oxygen preconditioning on the table of possible therapies. For now, none of these are proven or widely available. They illustrate how hungry the field is for neuroprotection beyond just lowering pressure. In summary, while these strategies sound promising in lab models, patients should remember that none are yet approved replacements for standard care. Lowering eye pressure remains the primary proven treatment for glaucoma, but these metabolic and mitochondrial approaches could one day become valuable add-ons to protect vision. Glaucoma and other neurodegenerative diseases. The concept of energy failure in glaucoma is not unique. In fact, it mirrors patterns in diseases like Alzheimer's and Parkinson's. In those disorders, aging neurons also lose NAD, mitochondria falter, and neuroinflammation runs rampant. Researchers point out that the same mitochondria inflammation feedback loop seen in glaucoma applies to Alzheimer's and Parkinson's. This means advances in one field can inform the other. For example, nicotinamide supplements have shown benefits in models of Alzheimer's and Parkinson's, hinting they tap into a universal neuroprotective pathway. Moreover, some genetic risk factors and tissue changes overlap. Optic nerve damage in glaucoma has been compared to the small nerve fiber loss in diabetic neuropathy or the brain atrophy in dementia. Scientists now talk about glaucoma more as a neurodegenerative optic neuropathy than just an eye pressure disease. This shift is useful. It opens the door to treatments developed for brain protection, like anti-inflammatory or metabolic drugs, and to broader lifestyle advice, exercise, diet, known to aid many neural conditions. Ultimately, tearing down the barrier between glaucoma and other neurodegenerations accelerates our understanding of both. Conclusion. In the story of glaucoma, the optic nerve is under siege on many fronts. High eye pressure, poor blood flow, and the wear and tear of aging all conspire to starve retinal ganglion cells of energy. When the cell's power plants, mitochondria, fail, a cascade of oxidative damage and even immune attacks follows. This appears to be a core part of how glaucoma kills vision. Science is now exploring therapies that target this energy crisis. Early research, from vitamin B3 supplements to antioxidant cocktails and gene tweaks, shows that bolstering cell metabolism can dramatically protect RGCs in animals. Small human studies hint at benefits, but larger trials are needed. For now, these ideas remain investigational. Patients should continue proven care, like pressure-lowering drops, and discuss any new supplement or therapy with their ophthalmologist. But it is an exciting time. The notion that glaucoma is partly an energy failure disease links it to all degenerative brain diseases, suggesting that future treatments might help preserve optic nerves just as they aim to protect memory or movement centers. In the meantime, a healthy lifestyle, good diet, exercise, blood sugar control can only help the optic nerve's fragile power systems. Continued research in this area promises not only new hope for glaucoma patients, but potentially for a range of neurodegenerative conditions as well. All links to sources are available in the text version of this article. You can find the full article at visualfieldtest.com. Thanks for listening. To check your visual field, click the link at the bottom of this article or visit visualfieldtest.com.